This presentation is a brief introduction to haemochromatosis and iron regulation, and I chose this topic because I feel like often there's a focus on what happens if you have too little iron in your body and the effects of iron deficiency anemia, but there isn't as much focus on the opposite end of that spectrum, which is what happens when you have too much iron in your body and haemochromatosis. To be able to fully understand the effects and symptoms of haemochromatosis, we're going to first go through a brief introduction to how iron is regulated in the body. As you might know, iron can exist in two ionic states: ferrous iron, which is Fe2+, or ferric iron, Fe3+. Now, to be able to be absorbed into the body, iron has to be in its Fe2+ form, or part of a ligand group known as a heme group. But after it's been absorbed through the small intestine, the body will often convert it into ferric iron, Fe3+, because it's less reactive and then will cause less damage. The reason why Fe2+ can be so damaging to tissues in the body is because it can react with hydrogen peroxide, which is a waste product from cells, to form hydroxyl radicals, and these are heavily oxidizing and they can cause quite a lot of oxidative stress to tissues and cells. Iron's journey through the body will start with its absorption through the small intestines, and from there it can go one of two ways. If there is current high demand for iron, then it can be attached to a protein known as transferrin and then transported to the area of demand to be used. This is usually places like the bone marrow, which is constantly producing hemoglobin and therefore has a high demand for iron. If not, it can be transported to cells to be stored inside a protein known as ferritin. And this diagram in the top right shows that. You can see that the ferritin forms almost like a shell or a coat around the iron, which is stored in its 3+ form as hydrated iron oxide, which is basically rust. From storage, it can be transported out of storage. Now, interestingly, there is only one molecule known to be able to transport iron out of storage, and that's called ferroportin. That's important later on, but for now, after being transported out of cell storage, it can be used. After the iron has been used in cells and the cells become senescent or disused, then the cells can be broken down by macrophages and the iron within them can be recycled to be stored again or used. You might notice that throughout this, apart from bleeding, which is listed, there isn't really a way to lose iron from the body, and that's true. Unlike other minerals like sodium or magnesium, where excess of it can be passed out with relative ease by the body in urine or feces, there doesn't really exist that mechanism for iron, which makes regulating it ever more important. A small amount of it will be lost as dead cells are excreted from the body, but apart from that, bleeding is the main source of iron loss. If you remember ferroportin, it is the only way to get iron out of cell storage. So therefore, to be able to control how much iron there is in the bloodstream and in cell storage, we have to control the amount of ferroportin there is available to the body. This is done by an enzyme called hepcidin, which combines ferroportin and begins its degradation, making it unsuitable for iron export. And this is an example of negative feedback. If there is too much iron in your body, in your bloodstream, then receptors on the liver will detect that, and that will stimulate the release of more hepcidin, which will bind to ferroportin, start its degradation, reducing the amount of ferroportin available to the body to export iron. Therefore, less iron is taken out of cell storage. Normal levels of iron in the blood will inhibit the release of hepcidin, which means that there is less ferroportin degradation, and that means there's normal amounts of iron being exported or taken out of cells. You might notice that this feedback loop also mentions inflammation, because another way in which iron is regulated is if inflammation is detected via inflammatory cytokines by the liver, then the liver will also start this feedback loop. The reason being is iron is used in quite a lot of important enzymes that regulate DNA replication and protein synthesis, and you don't want the iron to be able to be accessed by potential pathogens which are causing that inflammation. So haemochromatosis occurs when one of these steps of regulation fails and you get too much iron in your cell storage, in your body, or just in the bloodstream. And this is bad because the iron in your body will cause quite a lot of stress from oxidization and reduction. Haemochromatosis is usually diagnosed through blood serum levels, and a more than 45% saturation of iron on your transferrin, that is the protein that can transport iron, is said to be too much, too high. Haemochromatosis is almost always caused by a genetic mutation, and haemochromatosis caused by a genetic mutation is called primary haemochromatosis, primary iron overload. You'll see it's abbreviated HH. This is a medical abbreviation which will turn up later. Even within the bounds of genetic mutation, it's almost always caused by a mutation on a specific gene called HFE, and this type of primary haemochromatosis is type 1. It's very rarely caused by mutation on other genes, or even rarer caused by non-genetic reasons. The non-genetic iron overload is called secondary haemochromatosis. It affects roughly 0.2 to 0.3% of people in the world. This is significantly less than anemia, which affects around 25%. So there are 4 or sometimes 5 types of primary haemochromatosis. The first one is type 1 haemochromatosis, which is the mutation in the HFE gene, or the human homeostatic iron regulator protein gene. Now, HFE forms part of the iron receptor complex on cell walls, and it also assists in producing hepcidin, that enzyme which helps to break down ferroportin. The NHS website says that the condition of haemochromatosis will appear in a subset of people with 2 mutated HFE genes, and it's doing its best to simplify the condition for people who might be worried that they have it or just researching it. In reality, there are many different mutations that can occur on the HFE gene, and some of them require 1 mutated gene, some of them require 2 mutated genes, some of them can either just have 2 different mutated genes to cause symptoms to arise. Going back to our iron regulatory diagram, we can see that a mutation in the HFE gene will affect the detection of the iron by the liver, which means that there is not enough detection to sufficiently trigger the regulatory systems that keep iron in check. Type 2 haemochromatosis is split into type 2A and type 2B. Type 2A is a mutation on the HJV, the hemojuvelin gene, and hemojuvelin is an important protein that helps to stimulate the release of hepcidin or induce the release of hepcidin. Type 2B is a mutation on the gene coding for hepcidin itself. Now, while these two might not seem to have that much in common, they're grouped together because type 2A and type 2B both are early onset. Their symptoms will appear, unlike other forms of haemochromatosis, before the age of 30. And going back to our iron regulatory diagram, we can see that they both affect the release of hepcidin, which means that the body can't effectively regulate ferroportin levels. Type 3 haemochromatosis is a mutation on the gene that codes for transferrin receptor 2, or TFR2. And this is a receptor molecule on liver cells that can detect the level of iron in your blood via transferrin. And of course, like HFE, mutation on this gene will affect the detection of iron levels in the blood. Type 4 hemochromatosis is slightly different. Type 4 is a mutation on the gene that codes for ferroportin. And while the types we've seen so far affect the body in that too much iron is allowed to be in the bloodstream, type 4 affects the body in that too much iron is kept in storage. Iron export without ferroportin, iron export out of your cells is greatly reduced, and iron will start to build up in the cells of the body, especially in the liver where most of it's stored. While iron 3+ is less reactive than iron 2+, in great enough concentrations and amounts, it can still cause a bit of oxidative stress. In large amounts, which will be present in people with ferroportin disease, it can cause a lot of oxidative stress and damage to tissue. This diagram on the right here is actually liver cells under a microscope that have been stained with Prussian blue to show the presence of iron. You can see there's quite a lot of iron in the cells there. So, unlike the previous types that we've seen so far, type 4 hemochromatosis means that there's not enough export of iron, and so it just builds up in your cells. Now, type 5 hemochromatosis isn't really a separate type. It is digenic inheritance, where you inherit one or more different faulty genes in conjunction with mutation in the HFE gene, where maybe one of them on their own would not have been sufficient to cause symptoms to arise, but two of them compounding each other would lead you to have hemochromatosis. Now we know different types of primary hemochromatosis, we can move on to how they're treated. And it's through bloodletting. One of the main ways in which primary hemochromatosis is treated is through phlebotomy, which is the controlled bleeding to remove excess iron from the blood. Now, this will be done with scheduled appointments and regular checkups to remove small amounts of blood at a time, so that you're not draining your body of the blood that it needs. But it is still the modern cousin of medieval bloodletting, albeit with far more stringent hygiene standards. Of course, phlebotomy isn't a viable treatment in every situation, and often you'll need different therapies alongside it to treat different cases of hemochromatosis, especially with type 4, in which the damage to the tissue in the liver is so great that you may require a liver transplant. And for reasons that we'll come on to later, secondary hemochromatosis isn't usually treated with phlebotomy. Moving on to secondary hemochromatosis, it's quite difficult actually to get iron overload from non-genetic causes. To put the amount of iron in your body into perspective, the average adult human has about 4 grams of it at a time. This number will vary depending on if you're pregnant or breastfeeding, or obviously it will be slightly less for a child, but 4 grams is a reasonable threshold to set it at. The average human will consume about 200 milligrams of iron within their food every day. Out of this 200 milligrams, only around 2 milligrams can be absorbed by the small intestine. So you can see how difficult it is just by eating too much iron to get too much iron in your body. This is why secondary hemochromatosis usually occurs as a result of iron supplementary medication causing too much iron to be ingested, or from repeated blood transfusion to treat another condition. And this is where you can have anemia and hemochromatosis at the same time. If you have non-iron related anemia, because not all anemia is due to lack of iron. Anemia is merely the inability to have sufficient hemoglobin to supply all your red blood cells. If you have anemia and you are undergoing blood transfusion as a form of therapy or treatment for that, you are effectively introducing into your body quite a lot of iron. And from that, you can get haemochromatosis alongside your anemia. Now, to treat secondary haemochromatosis, especially when you've got the other underlying conditions, phlebotomy isn't used. You don't want to be taking blood out of the patient's body if they have anemia, because that will only compound the issue of them not having enough hemoglobin and red blood cells. Therefore, a common course of treatment in this case is iron chelation therapy. And this is where you're given chelating drugs, or drugs that can bind to iron in your body and allow them to be excreted. A common iron chelating drug used for this is defersirox, and of course there are other treatments available as well. You can also remove the underlying cause if it's from iron supplements by just not taking the iron supplements as well. Now, haemochromatosis doesn't affect all patients equally, and there are a variety of factors that can affect how quickly the onset or severity of symptoms can be. The four most common of these are age, sex, diet, and ancestry. Sex, because, well, females have their periods and they shed quite a lot of blood every month through that, which means that it's harder for too much iron to build up in their body when they're losing it that frequently. Of course, after menopause, symptoms can start appearing because you're no longer having that regular loss of blood. Diet can be a minor factor. As we said, it's quite difficult on diet alone to overdose on iron. But if you already have genetic haemochromatosis, primary haemochromatosis, and you are enjoying an iron-rich diet, then that can compound the effects and symptoms. Ancestry, well, obviously, as primary haemochromatosis is a term of genetics, there are certain populations that are more susceptible to inheriting faulty genes related to iron regulation. The people of Celtic background and origin in particular have a higher chance of inheriting the faulty gene just because of genetics alone. And age, well, the symptoms of haemochromatosis can take quite a long time to exhibit themselves. Indeed, it takes time for iron to deposit in your tissues and iron to build up around your body if you've got haemochromatosis. And so the symptoms, apart from type 2, will usually appear in old age, and often they are just put down to old age without further checkups, because iron deposition in your joints can manifest itself as painful arthritis, which is quite common in aging populations. Of course, you might be thinking, well, if it's just arthritis and inflammation, then surely it's not that much of a risk. But studies have shown that increased levels of iron in your body, haemochromatosis, can increase your likelihood of getting certain conditions, developing certain conditions. There's been studies to show that increased iron levels in your blood can increase your risk of cardiovascular disease. Firstly, because of the stress that the oxidative stress and inflammation that excess iron in your blood will cause to the tissues in your heart as well as your blood vessels. But the second reason is excess iron can deposit in fatty deposits in the arteries, making blockage more likely. There's still quite a lot of research and debate around whether high iron levels cause higher iron levels results in a higher risk of neurodegenerative disorders, or whether neurodegenerative or disorders exhibit higher iron levels as a symptom. But either way, iron chelation therapy has been shown to, in vitro, not in vivo, unfortunately, in vitro to have a positive impact on brain cells that have been modeled around brain cells in people that have neurodegenerative disorders. Of course, with higher iron levels in your body, you are more susceptible to more serious infection because there is more iron available for pathogens to access. We explained this as iron being an important mineral in making enzymes that control DNA replication and protein synthesis. If there's more iron available for bacteria or viruses to utilize when they infect a body, then of course their infection will be more severe. And of course you've got arthritis because of the iron deposition in your joints. So in conclusion, haemochromatosis is the presence of too much iron in your body, and this affects around 0.2% of people worldwide. Haemochromatosis can be genetic, and this is called primary haemochromatosis, or very rarely non-genetic, and this is called secondary haemochromatosis. The inability to regulate iron in your body can lead to stress to tissue via oxidation and reduction and inflammation, as well as iron deposition in various parts of your body. Most of the time, symptoms will start to appear more rapidly in old age, but there are types of haemochromatosis that exhibit symptoms in younger years as well, or early onset. Treatment can range from methods to remove iron from the body, like phlebotomy, to just removing the underlying causes, like stopping iron supplementary medication, or it can even be chelation therapy in some cases. The onset and severity of its symptoms can vary due to factors like age or sex. I hope you found that to be helpful and interesting.